Welcome by 111 human biology students. In this lecture video, I'm going to introduce you to chapter 1 in our human biology textbook. This is what it looks like. This is the main textbook we'll be using in this course. And you also will need to get the lab manual that looks like this. This is specifically for the BMCC students. Both of these books can be obtained through the BMCC web store online. Now, when we're going over any chapter in this book, you'll see that the author actually goes into a lot of the material in much more detail, or essentially spends more time on a certain topic per chapter. We're not going to be going over every single point that the author discusses in the book. We will primarily focus on the material that I believe is particularly relevant to us in this course. And so when you're reading the chapter, pay attention specifically to what's written in the class notes, which are on Blackboard, and what we'll go over in these video lectures. Now, to begin, essentially, chapter 1 introduces us to basics of biology and science. How do we do science? How do we deal with biological systems? How do we define living things in life? And kind of basic concepts like that, and easing us into the more complex topics of human biology, of the organ systems, and what's involved there. So we're going to be starting pretty slowly at the very beginning of the semester, going through some of these basics today, going over some chemistry in the next chapter, and kind of then again easing into the rest of the topics there to get ourselves better acquainted with the overall organism and how it functions and the structures that it contains. Now, for this chapter again, so part 1 here, essentially, what are the characteristics of life? When you think of something that is living, something that's a living organism, so we all know that essentially pretty much everything in the universe consists of atoms and molecules, right? We'll go into more of these details, what those means in the next chapter, if you haven't heard about this before, but essentially everything contains of different kinds of atoms and molecules. For living things, they also contain different kinds of molecules, but those molecules often are organic or living organism produced molecules. So whether something is living or non-living, in both cases they will contain atoms and molecules. And molecules essentially just multiple atoms combined together in a more complex form. So that feature doesn't yet let us know exactly whether we're dealing with a living thing, but we just have to keep in mind that yes, in fact, living things do contain that molecular composition. Now, much more importantly, close to the heart of the matter, is we have to realize that when you compare something living versus something non-living, like let's say a plant and a rock, even though both of those things will contain molecules, the rock does not need energy, does not need raw material, does not need metabolism to survive, right? The rock just sits there for millions of years, essentially does not care about anything, does not change in any way, does not need energy to live, to survive, right? Because it's a non-living matter. Now, so all living things, whether it's bacteria, plant, animal, ourselves, any animal, any living organism, from the smallest to the largest, will require energy, whether it's energy from sunlight, like from plants, energy from sugar molecules, like for animals, or other forms of energy, whether chemical or non-chemical. That energy is required to allow functions to take place in the body, in the organism. Metabolism is essentially a way to say that we take this energy and we pass it through a complex array of different molecular steps to get the product that we need to allow the cells, the organs to function. And very often, right, the organism also needs some kind of a raw material, some kind of molecule, some kind of elements from the environment in order to either make new molecules or survive. Now, as we're getting through this level of organization, keep in mind, right, so the smallest living unit in any organism is a cell, right? Once you go beyond the cell, right, you're just in the world of molecules and atoms. Once you combine this in the proper form, proper configuration, and you have a cell, that cell, even as one unicellular organism like in bacteria, is a living thing. And again, we'll be talking about cells quite a lot when we talk about microscopic anatomy, histology, and so on throughout the semester. So we'll be coming back at this more and more. Especially, we'll be paying attention to this topic in chapter 3 in the book. Now, from this point on, we're talking about essentially cells and overall more complex organisms, like ourselves, let's say. So all living things will require also a balance of their functions. This is what we call in biology homeostasis. Balance essentially means that we have to take in account that there's all kinds of processes taking place, sometimes good ones, sometimes bad ones. Sometimes the body or the organism generates waste that must be removed, right? We can't accumulate waste in cells or this will poison us. Sometimes we have too many of certain molecules. We have to get rid of them or balance out their presence. That kind of maintenance of balance, making sure all the functions are taking place properly, that there's not too much of something or too little of something, indicates to us the maintenance of balance and homeostasis in the body. Again, we'll be talking about this a lot throughout the semester and coming back to this topic. Also, very importantly, unlike that rock we mentioned before, that's non-living, obviously, all living things must respond to external environment. When you take a rock, now do something to it. I mean, you can say you broke the rock or you threw it into water, it's eroded by water, something else happened. But overall, it doesn't really affect the rock in any way, right? It doesn't change its identity. It doesn't do anything special to it. But living organism constantly must respond to external environment, right? If it's raining, you take your umbrella to make sure you don't get wet. If you're in the water, you have to know how to swim to survive, not to drown. If you're bacteria and you're somewhere on the surface, you have to make sure to hide or deal with that environment so you don't get destroyed by it. All these different ways, whether simple as a matter of using your senses to see where something is and run away from danger or deal with the enemies. Many examples of response to external environment exist, and I guess we'll be coming back to this throughout the semester as well in greater detail. Very important aspect of living things, again, response to external environment, right? We don't just sit there getting bombarded by all kinds of environmental things. We actually respond to that environment. Also keep in mind, right, very importantly, something I know that's very obvious but still needs to be mentioned, is that all living things, our cells, our organs, need to grow and reproduce. Even bacteria that is unicellular, one cell, still needs to grow to some extent, increase in mass, increase in volume, and respond to the environment and reproduce, split, and continue its genetic profile, right? Allow for reproduction. So all living things will be growing and reproducing to some extent, even if it happens just once in their life or multiple times, right? That is still very important consciousness, very unique to living things. Again, we'll be going over that later in the book as well in greater detail when we talk about the reproductive system towards the end of the semester. And now, not for a single organism, but for many organisms over many generations, over maybe millions of years or thousands of years or hundreds of years, we as species will evolve over many generations. Again, so talking about evolution, right? A lot of people are confused about evolution, not sure what that means. Essentially, this is change over a significant period of time, right? Thinking about the giraffes that early on had shorter necks and could not reach the tallest leaves on the tree, and then would die out because they could not eat that food and get enough nutrition, so had to grow longer necks to reach those leaves to be able to eat, and then passed on their successful genes, growing longer and longer necks and then evolving like that, right? But no giraffe obviously woke up one day and will all of a sudden with a long neck, right? It happened over extensive period of time, right? Something that's hard to kind of perceive for us because our lifetimes are very short, but in the grand scheme of things, right, over many generations, living things evolve. Now, part 2 here, let's talk about levels of organization, this kind of hierarchy that we have in science, right? This is referring to not just biology, but kind of overall talking about the universe. From the smallest, right, the smallest thing we have is an atom, right? Thinking about like oxygen in the air that we breathe, carbon, nitrogen, calcium, right? Any of those pure elements in one form as atoms, right? The smallest indivisible units. We combine them together, like H2O, hydrogen and oxygen, we get water, right? Now that we have a molecule. Then we combine carbon, oxygen, other elements, we get glucose, sugar molecule, right? There are many different atoms, there are many different kinds of molecules. Again, we'll go over this in more detail in chemistry, But essentially, right, as we start to combine them, if these are the kind of complex form that we need to make a cell, then eventually we get to that. We have a cell. Again, whether it's a plant cell, animal cell, bacterial cell, others, nevertheless, right, it has that composition of multiple different molecules that come together and work toward that uniform state of making the cell cohesive to make sure that it does properly its functions and survives. When we put multiple cells that work together in one uniform kind of structure, we get a tissue. So definition of tissue is a collection of cells working towards a common goal. Like you're looking at your skin and you're saying, okay, I have lots of cells in my skin. There are multiple layers of cells. These are different tissue types in the skin. Same thing for any other organ. Again, we'll go over this in histology or microscopic anatomy. When you put tissues together to make one organ, we get, again, an organ, right? So again, what is an organ? A collection of tissues working together towards a certain goal, right? So multiple tissues in your skin give you that organ, which is skin. Or multiple tissues, let's say, in your stomach or your lungs or your heart, give you that organ that's there. Now, when you combine different organs together that create one uniform kind of state, so organs towards, let's say, working for the cardiovascular system or respiratory system or others, you get an organ system. Okay, examples of organ systems would be digestive systems, right, for digestion, respiratory system for breathing, for respirations, cardiovascular for the heart and blood vessels, so on. When you combine all of this together, you get an organism, right, which I've mentioned already multiple times, right? That's your entire body, or an entire plant, or an entire even fungus. Any kind of organism will do here, right? As long as it has all its components. Obviously, for the most complex ones, like for us, for mammals, this is particularly important. We have that organism, and when you put multiple organisms together, you have a population of these organisms. And then further, right, we're increasing capacity to finally combining these populations. We have a community, and then finally ecosystem, right, where all the living things live, again, on our planet. For this course, we essentially are interested in the beginning aspects while we talk about chemistry, and then we'll spend a lot of time on cell, tissues, and the different organs and organ systems individually to talk about what they do, and we'll go over pretty much every single organ system in the body, going over the anatomy and sort of kind of basic physiology of those areas. For part 3, we want to talk about defining features of humans. So, what are the defining features? What are the unique kind of characteristics we can separate us essentially from other mammals, other kind of organisms or other living things, right? So you can say, so comparison to like primates, comparisons to monkeys, greater apes, things like that, right? The closest kind of genetically living things to us. So we share lots of characteristics, but certain things stand out as unique. Number one is the large brain. Right, so especially the frontal lobe in the brain, as we'll go talk about in the nervous system, right? So you can see kind of like in this book, this frontal kind of area is the largest. This is the one that has developed the most and basically allows for the complex functions of reasoning, personality, problem solving, motor control, and other types of control that is essential to make us unique humans, right? So we have the language processing centers in the frontal lobe as well. That's very important and kind of makes us stand out through that complexity. For the other primates, they also have obviously the brains and all the same kind of general features, but their frontal lobes are much smaller, and so they have fewer neurons there that generate that capacity, and so are not able to essentially generate language the same way and the same thought processes and the complex executive functions that are found in the frontal lobe. Again, so large brain, unique to us, especially relative to size, right? That's very important, right? So you're comparing other animals might have a larger brain, but their frontal lobes would be still smaller relative to their overall mass and have kind of a different connections and different processing centers there. The next part is the complex language, right? So it's not just vocalizations, but overall actually ability to produce language, to understand speech, very unique. So linguistics, language understanding, and production are very unique to humans. Obviously, other animals, they're able to produce vocalizations like some birds or other animals. It's all essentially not so much understanding, not a complex understanding of language, but just a vocalization. It's much, much simpler, requires a much simpler kind of brain structure to produce, right? And it's really overall should not be compared to human complex language abilities. Bipedalism, which is walking upright on 2 feet, 2 legs, right? So able to stand upright, see far the horizon. As we know, most mammals walk on all 4s, right? That gives them significant disadvantage in terms of seeing far, in terms of kind of ability to use tools, in terms of other kind of methods that related to early acquisition of skills and technology, things like that, right? So humans are unique in that sense. We are the only animals that are able to do that consistently, and our natural kind of position is bipedal state, and so gives us tremendous advantages in the world of, right, creating use of skills, use of the environment, use of technology, creating technology, things like that, right? Has led to significant and dramatic increase in that kind of potential in humans. And the last one, opposable thumbs, again, ability to use tools, right? When you think about when you're using your pen, that ability is very unique, very important. Other animals sometimes can kind of imitate this, but not to the same extent as humans, obviously, right? So that kind of a musculoskeletal system acquisition of the opposable thumb is very important and unique to humans. And the last part, the scientific method, very briefly. So we will be coming back to this in the lab and throughout the semester sometimes and in the book. Overall, there's kind of 5 steps here. As a scientist, when you come to work, you basically do your job very differently from other professions, other disciplines. As a scientist, right, you first, step 1, you observe and generalize. So as an example, for instance, let's say you have a classroom of students and you look at them and you've noticed that a few of the students are unable to see what's on the board, so they're squinting, they are sitting in different parts of the classroom but still cannot see the board, what the professor has written. So first, again, you observe these students and you generalize. You're thinking about, okay, so something is happening. Why can't they see the board well? Step 2, you formulate a hypothesis. And you're saying, okay, so it's just basically a question you're asking. Why can't some students see the board well? Why are they squinting? Why are they having difficulty seeing what's been written on the board? Step 3, you make a testable prediction. Let's say a prediction is, if I give to the students, let's say, a certain medication that will somehow improve their vision, will that have an effect? Will it make them much better, much more easier able to see the board? You conduct an experiment, right? So there's many different experiments. Obviously, this is just as an example. I'm giving you this. So like in pharmaceutical industry, when medications are created, we often want to have a double-blind, placebo-controlled study. Double-blind means neither the people administering, neither the people taking the medication know exactly who received what, to eliminate the chances of bias, or at least decrease bias. And bias is something that could be even kind of unconscious or semi-conscious, and it's very hard to kind of eliminate unless you really do this very strictly controlled trials. And someone must receive a placebo. Placebo essentially is a sugar pill, something that doesn't have an effect, but often still generates some kind of effect if a person thinks that they're getting the correct kind of treatment that they're desiring. Right? When you do an experiment like you would do normally in a lab, you have variables and controls. You have positive and negative controls. Positive control is where you have something where you know it always will come out positive. So for instance, if you're testing for sugar, you have a container that's is filled with sugar, obviously this is your positive control because it will always come out positive. And negative control is something you know for sure will always come out negative. So again, in that same test, if you're testing for sugar and you have a bottle of plain water that obviously is like, say, like tap water that normally would not have sugar in it, then this would for sure be a negative control because you know it always would test negative. And variables obviously could vary. Variables could be time dependent or dependent or independent variables depending on different scenarios of the experiments. Again, we'll go into more of these details in different experiments and different procedures and throughout the semester. And then the last part, we modify the hypothesis based on the results we got, based on the experiments. We repeat the steps and draw different conclusions.